So given that we want a primer to satisfy all these qualities that I've listed here, sometimes it may not be possible to design primers exactly at the start and end of the gene, and instead you may end up designing the primer somewhere in the flanks. For example, let's say here, right? So now you have your gene of interest, which will be amplified, of course, but then there is a bit of an extra sequence also that gets amplified along with the gene of interest. So as I said, the art of finding the most optimum primer pairs to amplify the target is known as primer designing, and is now done using primer designing softwares, right? And I've already told you the most common software that we use is Primer 3. It is also available as a web-based graphic user interface, and also it can be downloaded and installed for a standalone version, which can work on command line. So what we're going to now do is to look at why exponential amplification cannot happen if you have a single-stranded DNA and one primer to do your polymerase chain reaction. So here you are. This is your single-stranded DNA that you have, and you have a single primer which is complementary to the end of this template DNA. And you start with first round of your PCR, and the arrow again represents the 3' end, and the blunt end represents the 5' end here. And now when you start with the PCR, so the primer hybridizes to the template DNA strand, which is single-stranded here, and then the DNA polymerase extends it, so you have your extension. Finally, you have a double-stranded DNA here, which is then denatured in the next cycle. So we are now into cycle 2. This gets denatured. And after denaturation, again, the primer can hybridize only to the blue strand here. So it goes and hybridizes to the blue strand, and then gives you an extension, giving you the second complementary strand. And then again, if you move on to the third cycle, so you denature it. And after denaturation, again, you have your 3 DNA strands, 2 of which are complementary. One was the original DNA strand, and again, the primer hybridizes to the original DNA strand because it is only complementary to the blue strand and gives you amplification again. So if you look at the analysis of these extensions that you get, after the first cycle, you have 1 red strand. After the second cycle, you have 2 red strands, and after the third cycle, you have 3 red strands. And there is only 1 double-stranded DNA after the completion of the cycle. So therefore, this is not what is exponential amplification. And therefore, in cases where you have a substrate that is single-stranded, for example, the mRNA, it is first converted into cDNA, and then the cDNA, which is double-stranded, is then taken forward for amplification, right? So here you are. Exponential amplification does not take place in PCR with single-stranded nucleic acid molecule and 1 complementary primer, and I've shown you how this works out here in this example here. For downstream experiments, the RNA is first converted to cDNA for 2 reasons. Number 1, RNA is extremely unstable, and therefore, if you want to perform any experiment on RNA, it has to be first converted to cDNA, which is a lot more stable. And one of the reasons why the genetic material switched from RNA, which was largely believed to be the original genetic material, to DNA was the stability of the DNA. And then, of course, the second important point is that exponential amplification can only happen when you have a double-stranded DNA and 2 primers to start your PCR reaction. So therefore, when you work with RNA, first the RNA has to be converted to cDNA, and after it is converted to cDNA, it is then taken forward for quantification using exponential amplification. So this is what we're going to see next. There are 2 types of polymerase chain reactions that we're going to talk of in the next slide. One is what is known as reverse transcriptase Pcr, where we are going to convert the mRNA sequence into the complementary DNA sequence, and this complementary DNA sequence can then be amplified. And while amplifying, you want to see real time what is the level of amplification, which is basically done by what is known as real time PCR. So we're going to see how this works out in the next coming slides.